Okay. Which means so your date and time. So you walked in a little late, so now your fate is sealed. No, you're going to be the second presenter on December 2nd. So let me write this down, then I'll give you this little paper. December 2nd. There you go. You're welcome. Were your travels good?
Yeah.
Good to hear. Okay. Let's see. Nothing. I'm not laughing at you. I'm just like trying to give Josh more of a hard time just because.
That's okay. You can take it out.
Oh gosh. Okay.
Got it.
So, let's see, just to remind everyone, so, the trouble you're causing now?
Oh, I was going around and saying that I wasn't bullied endlessly in my youth to not be able to take it as an adult.
Oh, okay.
You're still bullied.
Okay, so, quick reminder, oh my gosh, the graduate students are gonna be taking over the class when we come back from Thanksgiving. So, in a good way. Don't look so scared, Peter. You're gonna be fine, okay? All good. Let's see, so, and I will actually post who's giving what, or actually, graduate students, please, over Thanksgiving break, send me the title of your presentations. And that way, what I can do is to essentially make a nice spreadsheet of when people are giving their presentations in the titles. And so I can post that on Canvas so that the rest of the class is informed.
So if I wanted to type a one-page summary of what my presentation was going to be about and submit that with my presentation, can you put that on the Canvas as well, just in case if someone, say, gets sick or has a family emergency the day that I can?
Okay, I'm going to tell you one better. Okay, so the day of your presentations, okay, actually before you come to class, on the day of your presentations, you need to email me 2 things. 1, email me your questions for potentially being on the next exam. And again, my qualifier is that I get to edit them however I want to. And then 2, provide me your PowerPoint presentation. And that way people have the slides, and if something does come up or whatever, then they can get notes from somebody else in class, or even email the presenter and just ask, hey, what do you mean by this type of thing? Okay, so Jeffrey, since you're our first guinea pig, you'll need to email me your presentation and your questions, Monday before we meet for class. So, yeah.
Is the paper part due the same way or no?
No. Okay, so the reason why the papers are actually due by 5 p.m. the last day of finals week. The reason for that is because what's going to happen is as you're giving your presentations, there's going to be good questions because we have a very intelligent group of people here. They'll ask, except for maybe Brian. No, you set yourself up, Brian. Brian is also very intelligent too. But we have a very intelligent group of people. They're going to ask smart questions. And so that might actually lead you to want to revise part of your paper. So I want to give you the very best opportunity to submit your best efforts. So that's why the papers are due by 5 p.m. the last day of finals week.
Yeah. How long is the presentation supposed to be?
Plan for about 10 minutes. 5 minutes for discussion. And typically my rule of thumb is that about a slide per minute. So it'd be fine to have between like 10 and 15 slides. Just don't come in with a deck of like 30 or more slides. Please do not do that. Okay, because I'll go, or actually I'll set my timer, so.
We're shooting for what, 15 minutes for presentation and then
Pretty
5 minutes for questions roughly.
No, Josh, you should have been listening.
I got a lot to think about right now. Okay, my brain's in like 3 different places trying to think about those. As you were telling me, now I gotta
For
go
real
dig
life.
a hole in the environment to go find these microbes, so I gotta think about how I'm gonna do that.
That's right.
Wasn't in my plan.
Not too bad.
That's why I wasn't listening. I'm sorry.
Okay, so like I said, aim for about 10 minutes presentation. But, you know, if you go over, it's okay. So essentially, full time for the presentation, 15 minutes for, 5 minutes for questions and answers. But the nice thing about only having like 1 presentation to 2 presentations a day, we can go a little bit over, but we plan for about 10 minutes uninterrupted talking, except for Josh. Go ahead and interrupt Josh.
No, ask questions with my presentation. I will give you the candid response of I don't know and continue on.
Or you could just say,
Okay.
your questions.
See, there you go. Laura, good.
That's making me do something I actually know how to answer that.
Okay.
You gotta keep Josh.
So, okay, any other questions? So, okay, let's go ahead and continue on with biogeochemical cycling. So we covered carbon cycling and we started to discuss nitrogen. Woohoo! Okay. The nitrogen cycle, like I said, what we're really focused on actually is pretty much the cycling of the inorganic forms of nitrogen, as well as the nitrogen associated with biomass, which is going to be ammonia-based, really. So we started to first talk about nitrogen fixation. And so what are some of the key points about nitrogen fixation?
You pointed to one of the nitrogen symbols on the chart there and had an R and a hyphen.
Right.
Is that are those the nitrogen or tested proteins?
This symbolizes any organic
Oh,
moiety.
gotcha.
So, yep, you're welcome. Okay, getting back to nitrogen fixation, what are some of the big overall points about nitrogen fixation?
Has to be under aerobic conditions.
It has to be done under anaerobic conditions because oxygen will destroy the enzyme responsible for nitrogen fixation. Okay, and what else?
Most of the organisms are aerobic organisms, but they need to find some way to create a separate environment to run we were talking about the rhizobia, and then also there's the cyanobacteria that form the heterocysts. They find a separate way
Okay,
to
so
create an
essentially
anaerobic
they have to
environment
make
within
sure,
themselves.
right? Okay, and then what else? And actually thinking about this wash process. So humans have been trying to replicate this because essentially when you think about the elements that are limiting for most biomass production, it's going to be nitrogen and phosphorus. And so why have people put so much effort into producing industrial fertilization, and why are people concerned really about, these processes and trying to find better ways of producing? Josh?
They're incredibly energy intensive and require a lot of materials to actually work.
So it's incredibly energy intense. And for the abiotic pressure, so you have to have high temperatures, ergo lots of energy, fossil fuel being used for this, and high pressures. Again, lots of fossil fuel has to be invested. But when we think about the microbial cells, okay, for every molecule of dinitrogen that is fixed to ammonia, the cell has to invest 16 ATPs. So it's very high energy for the organisms too, but microorganisms do this at regular pressures and temperatures. And so people are trying to figure out how to do that abiotically to save on fossil fuel. Get your hand up.
Yeah, I was looking it up and I Googled it, but it came up as Haber-Bosch, but then I was is that the same one?
It's the same.
Okay.
Yep. So, and again, the name of the process isn't really that important for this class, but
No, but when I
pressure-
read about it later, I want to, got it.
Right. Yep. Yeah, Josh.
Well, so you said that they do it at normal pressures. Is that because they have the enzyme that enables them to do it at normal pressures? Whereas, like, since we're just trying to separate it out, we actually break it without any sort of like.
We don't, like I said, with the enzyme being so viable, essentially fragile. So that's why we just can't go ahead and, extract this enzyme and utilize it. So although I would suspect that there's probably some biotech companies, startups that are specifically looking at things like that, but okay. So nitrogen fixation. And nitrogen fixation is also one of these processes, except for that abiotic process that humans do, only bacteria are capable of doing nitrogen fixation. Okay, we started to just mention about nitrification. And so with nitrification, essentially what we're doing is, so nitrification is totally aerobic, okay? And actually one of the major applications of this process is wastewater treatment. And so you have things like trickling filters and rotating filters and all that allows a biomass of these bacteria to grow. And so it's a two-step process. And so when you think about wastewater, and a way to also remember how this process begins, it's weird seeing my hair up on the screen there. But, when you think about, the form of nitrogen that ends up in wastewater, it's going to be urea, ammonia, okay? And so that is very reduced, all those hydrogens. And so you have this aerobic process in which you first take the ammonia and it's oxidized to nitrite. Okay, so let me write that. Okay, so it's important to distinguish between nitrite and nitrate. And so it's a two-step process in which you have organisms oxidizing the ammonia first to nitrite, and then you have another set of organisms that takes the nitrite and oxidizes it further to nitrate, so to NO3. And like I said, it's used in wastewater treatment. It's actually a very old process that people have relied on for centuries to clean up wastewater. Now, some of the things, as noted, and some of the problems that wastewater treatment plants can get into is if you have a disruption in these microorganisms, then it can all shut down. So these organisms are very sensitive, and especially if the pH gets too low, they'll shut them down. So, okay. So now going from nitrate, and again, here's one of these inorganic forms of nitrogen. In fact, the most oxidized form of nitrogen, nitrate, and actually one of the easiest or one of the common forms found in the environment for organisms to utilize. Well, essentially, organisms are not able to just take the nitrate and directly produce biomass molecules with that. The nitrate has to be reduced. And so when you are actually forming biomass, the term that's used is assimilatory. So this is assimilatory nitrate reduction. And one of the ways that you can distinguish between nitrate, and we'll talk about dissimilatory reactions in just a moment. Okay, so with assimilatory, those products are truly going to be assimilated or built into biomass. Okay, and so one of the ways to sort of remember that, and some of you guys have heard me say this before, so don't raise your hand, Josh, but who have heard of the Borg?
It's a tennis player.
Huh?
It's a tennis player.
The what?
Said he's a tennis player, isn't he?
Oh, no, okay, that's really going back, beyond Borg. No, not him, but the Borg in Star Trek. Okay, so. But
I got you.
Thank you. I know, I'm so glad that I was able to connect with somebody besides Josh. Yeah.
Make it sound like a bad thing.
It is a bad thing.
Okay,
Alright.
no, stop it. But, no, so some of although for Star Trek, and their thing is that they would assimilate and bring in all these other cultures that would then become Borg. So, but Miranda gets it. Thank you, Miranda. And Josh should get it because he's heard this before. So, which is why I did not. Except for a little bit. I know, because I can. But with the assimilatory nitrate reduction, what you're doing is you're going through the process of reducing the nitrate and forming ammonia. And the ammonium is then introduced into biomass, actually through the formation of an amino acid glutamine. But essentially, this ammonia is how the nitrogen actually becomes part of biomass. Now, when we have dissimilatory nitrate reduction, with dissimilatory reactions, okay, this is where you're actually using one of these inorganic molecules as a terminal electron acceptor. And so essentially, the electrons are getting dumped onto nitrates as a form of respiration. And then what happens is that this does not become incorporated into biomass. It is truly going to be a waste product. Okay, so it's not made into biomass. And so with the dissimilatory nitrate reduction, what you're doing is you're going from nitrate to nitrite. And one of the things that can happen is you can have this nitrate ammonification. So again, what you're doing in that particular case, with the dissimilatory nitrate to reduction to ammonia, you're forming the ammonia as the waste product. Another version of this terminal electron acceptor process is instead of reducing the nitrate to ammonia, you actually have these additional steps going to dinitrogen gas. And so that process is denitrification. So denitrification is when another form of the terminal electron acceptor utilizing nitrate, but you also have all of these steps that enable the organism to shunt their electrons to those intermediates.
So we're using every step of the way as a terminal electron acceptor from nitrate all the way to
Dinitrogen.
So
Okay, so essentially, no. What you're doing for the denitrification is it stops at dinitrogen gas. And then that dinitrogen gas is released into the environment.
but we're using everything up to the dinitrogen monoxide as a terminal electron acceptor, right? And then at that point, when it goes from N2O into N2, then it's no longer a system.
Right.
Okay.
Exactly. Any other questions about this? Yes, this is a bit of, memorization. So, okay, so now one of the interesting things that can happen is that you can have ammonia oxidation under anaerobic conditions. Okay, so we talked about the nitrification earlier, in which organisms have to have oxygen. However, under anaerobic conditions, nitrate can be used as a terminal electron acceptor and accept those electrons from the ammonia. And so this goes through and you get the final product of nitrogen gas. And one of the interesting things that have come out and why people are really intensely studying this is its useful for wastewater treatment. And the reason for that, like I was telling you about the nitrification process and all, okay, has to occur under strictly aerobic conditions. We didn't actually go over the specific organisms that are required for this process, but they tend to be very sensitive and they can be disrupted. And that whole process of providing enough oxygen and all is very energy intensive. Something like 1% of all of the electricity usage in the United States is specifically for the nitrification process. So when you think about it, that's a huge amount of energy. Potentially fossil fuels that are being utilized to clean up our water. So if you can do a process in which you don't have to have strict aerobic conditions because it's really providing the oxygen that's taking up that energy, then you can reduce energy usage and costs. And so that's why people are studying this, actually, Dr. Jianmin Wang. And and that's why I know off the top of my head the 1% because I served on a couple of the students' committees. Yeah. So yes. And the thing is that if these things are implemented more, it can be a huge cost savings and potential reduction in fossil fuel use. So, he's got a winner in me, but no, actually, it's his data, his savings. Yes. Right. It's one of the things that he has specifically looked at is when you have well, especially like military deployments, being able to quickly bring out, methods to treat the wastewater and everything that's going to be produced is going to be incredibly beneficial. So, yeah, sure.
Is this having, does it happen?
Yeah, because one of the next things I was going to mention is that in our own lab, we're actually Cassie Roberts is specifically looking at, she's teasing out the different nitrogen metabolisms that's occurring in this environment that's actively going through bioremediation. And so this actually might be a key step, but I actually anticipate pretty good stuff. So, okay. So let's see. Any other questions about nitrogen or any different processes and stakeholders?
So what is the ammonium oxidized into? It's oxidized into
It's
nitrate,
actually-
right?
Okay, so the ammonia is, so the nitrite has to be present. So you have to have ammonia and nitrite. And the electrons from the ammonia being transferred to the nitrite, and then they're both going to be reduced, or actually, reduced to
So
gas.
that's the whole thing.
Yeah.
So but the ammonium doesn't just go straight to that nitrogen, right? It
No.
turns into nitrate, but
Well,
when we look at that giant
Yeah.
flow chart, it goes nitrate, then the nitrite, and then it goes to, okay, got it.
So, but the initial step is the use of nitrite. So, and actually, that's one of the things that, towards the middle of next semester, Cassie is going to actually have a nice diagram produced in which she shows where all these regulatory quality electrons. So she sort of finished that a little bit, but she's been really doing this. So all of the electrons. So, hey, anything else about nitrogen? Sure.
We found that nitrogen fixation. And I see two different
Yes.
So how does nitrogen fixation and nitrogen?
Okay, so with nitrification, what you're doing is it's, with the nitrification process, that's this process here. Okay, the nitrogen fixation, this is where you're taking dinitrogen gas, two nitrogens triple bonded, breaking those triple bonds, and then reducing each of those nitrogens to ammonia. So,
But it's the bacteria that's doing this, right?
oh, they're
It's
different bacteria.
different bacteria.
Very different bacteria. Yeah, so, and actually getting back to Cassie's project, we're hoping to get funding so that we can do metagenomics to really, so Cassie's got really good data. And now what we want to do is have that level of reassurance that we have exquisite microorganisms are present that are capable of doing these metabolisms. So they're different organisms.
Yes, so it has like the particular of the cyanobacteria can perform this key process.
Okay, cyanobacteria can form nitrogen fixation. It will not be able to do this because cyanobacteria are, for the most part, strictly aerobic. So in this particular process is anaerobic. This process is strictly aerobic, but it's actually very specialized organisms that do this, not cyanobacteria. Okay. So let's now turn our attention to sulfur. Okay, so where do we find the most sulfur? is going to be black again. But the ocean also has a large reservoir of sulfur in the form of sulfate. That's what we're talking about, the salts of the ocean and stuff like that. The atmosphere actually does have quite a bit of sulfur sulfate, or sulfur oxides and hydrogen sulfide. And actually, you took my microbiology class. No, retreat. It's next semester. Okay. So we can't pick on you, darn it. And looking around, I think checkpoint analysis that might microbiology yet. Okay, so I take microbiology. It's awesome. But one of the things, if you see highly oxidized compounds along with the highly reduced analogs, that is actually a signature for life. Because what happens abiotically is that you're going to get a equilibrium occurring, and this actually shows that there's a disequilibrium of these compounds. So it is exciting. So you can see stuff like that in other planetary bodies. Awesome.
I hate that I'm going to go down this rabbit hole. Equal, no life, disproportionate life.
Okay, so essentially what you tend to find is that, like, well, so you tend to will find one or the other. Yeah. So, but if you find the presence of both oxidized and reduced, like Earth, not only do we have this, with the sulfur, but also with carbon, we have this concentration of carbon dioxide and methane in our atmosphere that is not in equilibrium. And so Carl Sagan actually noted that as, signature of life. So Josh, you mentioned so that oh, okay. Okay, so let's consider the sulfur cycle. And actually, sulfur cycle is pretty much my introduction to anaerobic microbiology, because I was interested in studying sulfur-producing bacteria and all for a while. But really, with the sulfur cycle, it's again going to be taking into consideration the organic form, inorganic forms of sulfur, balancing between anaerobic processes and aerobic processes. And then we've also got, again, a simulatory sulfate reduction occurring that's essentially going to introduce sulfur into biomass. So we'll go through there. So let's first consider that as simulatory sulfate reduction. And so what we have is that, so sulfate is not toxic. Hydrogen sulfide is, but the form that life actually uses to incorporate, when you think about it, and I'll call on my biology majors, but name a amino acid that contains sulfur. Cysteine, thank you. And so what form of sulfur is that cysteine? It's going to be a hydrogen, H. Yeah, so SH. So the sulfur, I'm sorry, is that methionine? Yeah, methionine is the other one. And so it's sulfides that, for the most part, make up the sulfur-containing biomolecules. But the thing is hydrogen sulfide is very corrosive, very reactive, very toxic. And so the cells actually do have to use a much less toxic form of sulfur, and that's in the form of sulfate. So with assimilatory sulfate reduction, you go through the steps of reducing sulfate to the sulfide moiety. And again, the R stands for organic compound. So, and then what occurs, so they've got the sulfur mineralization. And so with all of these, when we do have the degradation of biomass and all, this is going to be further reduced to form hydrogen sulfide. So now, to essentially replenish the sources of sulfate, what we have is the process of sulfur oxidation. Okay, and so this is going to be an aerobic process. So it has to occur under oxygen conditions. But your first step, and this doesn't necessarily have to be under aerobic conditions, but essentially you have the oxidation of hydrogen sulfide to elemental sulfur. And so, actually, jumping ahead, there's trophic oxidation anaerobically, but you have this occurring under strictly aerobic conditions and formation of elemental sulfur, and then you can have a further oxidation of that elemental sulfur to sulfate. Yeah, one of the things to also highlight, and we talked about this, when we talked about gasifying drainage and all, is that when we do have, especially metals that contain sulfide, and you have the introduction of oxygen, those sulfides will be oxidized, forming sulfate, and what's another form that sulfate is? Sulfuric acid. Okay. So, okay, so what I was saying before about how you can also have these things that are occurring anaerobically. Okay, you can have anaerobic photosynthesis. Okay, and so when we have anaerobic photosynthesis, what's occurring is that these anaerobic photosynthesizing organisms are essentially using the hydrogen sulfide as their source of electrons. So when we have oxygenic photosynthesis, what actually is the compound that's used by oxygenic photosynthesizers? We just talk about photosynthesis, like if there were still leaves on the trees, they're going to take carbon dioxide and fix that, reduce the carbon dioxide with the electrons from this compound. But what is the compound that aerobic photosynthesizers use for their electrons? Is what is compound that would provide, electrons. Oxygen is the waste product of photosynthesis. Water, yes. So, yep. So aerobic photosynthesis relies on water as the source of electrons. Okay. On the other hand, anaerobic photosynthesis relies on hydrogen sulfide. And so you have the removal of the electrons, okay, so it's being oxidized to elemental sulfur. And actually in some of these places where you do have a lot of anaerobic photosynthesis occurring, you can actually see the deposition of elemental sulfur. Okay, but then there's other organisms that can further take this and continue to oxidize it to sulfate. So this is some of the sulfur oxidizing bacteria that are present. So, like we were saying before about this line drainage and stuff like that. Some of the different organisms, and then anaerobic phototrophs. So, and essentially the areas where you find those organisms. Okay. Now, with dissimilatory nitrate or sulfate reduction, okay, again, what we've got is organisms that are using sulfate as their terminal electron receptor. No, because that's just, it's the type of thing, it'd be good to know, but there's so much material. So I'd much rather you focus on the processes. So, okay, so with sulfate, dissimilatory sulfate reduction, okay, what's occurring here is that these microorganisms are using sulfate to essentially breathe. And again, shunting the electrons from metabolism to sulfate. And you're not showing all the steps, but there's a sulfite step here, don't worry about it. But this will be completely reduced to hydrogen sulfide. And so when you do have an environment where you have a lot of sulfate reducing bacteria, you're going to have a lot of hydrogen sulfide. And no, it was actually a guest speaker on Monday, but the different aromatic lakes that are out there. So aromatic, if you remember, those are the lakes that don't turn over. And so Soap Lake, which is an aromatic lake, which we have studied, we have an awful lot of sulfate reducing bacteria there, and the deposition of hydrogen sulfide in the bottom layer of the lake. And actually, our group was the group that actually measured the highest naturally occurring sulfide concentration from that lake. So, our group in public with the Russian scientists about this. I'm so proud of myself. So, and I won. Oh, I got a nice apology. No, I'm serious because of world events.
Oh,
Yes.
you sound like Don Corleone right there. Like I argued with him and I won. I hope it's okay.
I'm of Italian descent. Okay, but watch it. Okay. Any questions about sulfur before we go on to iron real quickly? Huh? Oh, iron, yes. Okay, so iron. Okay, so again, what? Okay, so and again, when you think about where iron is mainly going to be found, it's going to be pretty much in deposits and stuff like that. Okay, now what we've got is iron is funky, and actually this is something that's actually causing gassing to have lots of debates right now, because not only do we need to consider okay, so then actually again we become Josh, since he's had his my biogeochemistry course, or wait, sorry, my geo micro course. Boom. Okay, so what form of iron is that?
That's the one that's less soluble. I don't remember what I'm thinking of. I think it's ferric iron.
There you go. See? Yeah. So essentially, ferric iron is like rust. This is the oxidized form of iron. The reduced form of iron is ferrous, so Fe2. And so, organisms can essentially, iron reducers can take the ferric iron and form ferrous iron. Iron oxidizers go the other way. But on top of this is that you can have different crystalline structures. Okay, and so that's what they're showing at the top of this slide. So we have our ferric iron, so ferric oxide continuum. And one of the things to think about is that if you've got iron reducing bacteria, so just iron reducers. Okay, so if you are an iron reducing bacteria, are you going to preferentially be able to reduce like an amorphous, sort of a flocculent type of iron, or a highly structured crystalline one?
That one, the second.
Okay. I think you might have an opposing point of view.
I would think not the crystalline one because that would be like a harder structure to.
Yes, exactly. So essentially, what's going on here, and when you do see like the more amorphous form and all, you don't have that crystalline structure that you actually have to invest energy into to break the crystalline structure. Okay, so that's why you have this continuum. So if you do have something that tends to be very structured, like earthite, it can be reduced, but it's going to be harder for the organisms to shunt those electrons to it. Okay, so that's why the continuum up here. And so you have the aerobic areas where you do have the ferric iron. You have this interface, which is what they're indicating here. And so you can have the ferrous. The ferrous iron is going to be more soluble than ferric. And unfortunately, we don't have enough time to talk about banded iron. Look up the term banded iron formations. But essentially, the banded iron formations occurred as Earth went from being a reduced environment to an oxidized environment because of those phototropes. So phototropes caused the greatest pollution event on Earth. And the result was the banded iron formation. But other things to keep in mind about the iron cycle, and again, getting back to iron that can be utilized for biomass. Okay, so preferentially, the organisms would want to be able to take up the soluble form of iron, ferrous iron. However, most of the iron is going to be in the less soluble form, ferric. And so organisms produce these compounds, these siderophores. Okay, so that's SIDEROPHORES. but these are specialized molecules that organisms produce that scavenge ferric iron from the environment and are able then to take iron into the cells to assimilate it. Okay, so organisms need those siderophores to bring in iron for their biomass. Now, so any questions? Yeah.